Students, already we have seen alkylating agents and antimetabolites under anti-neoplastic agents. So, in this class we are going to see antibiotics that are used in the treatment of cancer. These antibiotics, they are classified into 5 types. They are anthracyclines, bleomycin, mitomycin, dactinomycin and plicamycin. Under anthracyclines, examples are Daunorubicin, doxorubicin, idarubicin and carminomycin. Under bleomycin, only one, that is bleomycin sulfate. Under mitomycin, mitomycin C. Under dactinomycin, that is called actinomycin D. And the last one is plicamycin. Under that, example is mithramycin. This is the general structure of anthracyclines. So, what it is having? It is having a glycone and this one is called glycone, and that two are connected by means of glycosidic linkage. Already we have seen examples. What are they? Daunorubicin, doxorubicin, idarubicin and carminomycin. In the case of Daunorubicin, we have, that is methoxy group that is attached in the fourth position, and here we have CH3. So, that is for Daunorubicin. In the case of doxorubicin, same as that of that, here one hydrogen is replaced by means of a hydroxy group. In the case of idarubicin, here we have only hydrogen, but here we have CH2OH, that is for idarubicin. And in the case of carminomycin, here we have CH3 and here we have hydroxy group. So, that is for carminomycin. And this one we have seen A-glycone, that can be called as anthracyclinone derivative. So, this is an anthracyclinone derivative. And this glycosidic linkage, how it is forming? Here we have hydroxy group in the 7th position. So, 7th hydroxy group of anthracyclinone binds with the beta anomer of sugar with L configuration. So, that is the glycosidic linkage present here. Now, and in the case of Daunorubicin, so what is for Daunorubicin? Already we said in the 4th position we have methoxy group, and here we have CH3, that is for Daunorubicin. So, what is the glycone present here? That is called as danosamine, L-danosamine. This is called L-danosamine and this is called as danomycinone. So, this part that is called danomycinone and it is an anthracyclinone derivative. This doxorubicin, compare these two, Daunorubicin and doxorubicin. Here we have hydroxy group, in that position we have hydroxy group, CH2OH. So, it is a 14 hydroxy. So, it is a 14 hydroxy analog of Daunorubicin. The anthracyclines are isolated from Streptomyces caeruleo and they are the glycosides of anthracyclinones. Here already we have seen that how the glycosidic linkage is formed, that is between the 7th hydroxy group of anthracyclinone and beta anomer of the sugar with L configuration. So, what is the A-glycone present here? That is called as anthraquinone derivative. What are the uses? That is used in the treatment of bronchogenic carcinoma, acute lymphocytic and granulocytic leukemia. This Daunorubicin, here the glycoside that is formed between danomycinone with L-danosamine. While comparing Daunorubicin with the doxorubicin, doxorubicin is a 14 hydroxy analog of Daunorubicin. Now, we will see the mechanism of action of anthracyclines. The anthracyclines, first they are intercalated, that is inserted into the double helical DNA. Then they inhibit the enzyme called topoisomerase II. After inhibiting that enzyme, it will inhibit both the DNA and RNA synthesis, and it also generates ROS, that is reactive oxygen species. Already we have come across that. What are they? Superoxide radical anion, hydrogen peroxide and hydroxy radical. They produce destructive effects and leads to damage the DNA. Next drug is called as bleomycin sulfate. This is the structure of the whole thing, that will be the structure of bleomycin sulfate. So, it is having a pyrimidine nucleus and that is having amino group, methyl group, and here that is having ketone, and here we have that is having beta amino alanine group. And with the ketone, what is attached? Methyl valerate is attached, and that is attached with the threonine, and that is with 2 thiazoles. 2 thiazoles would be attached, and here with this carbon, what is attached? one more carbon, and with the carbon imidazole, and here that would be glycosidic linkage, and that is having two sugars, one is alpha L glucose, another one is alpha D mannose. This bleomycin sulfate, no, that is having two types, one is bleomycin A2 and another one is bleomycin B2. One more form that is called as bleomycinic acid. In the case of all, we have hydroxy group means that is called as bleomycinic acid, and in the case of all we have NH thrice, SCH3 twice means that is called as bleomycin A2, and in the case of R we have NH 4 times, NHC NH2 double bond NH means that is called as bleomycin B2. This bleomycin is isolated from Streptomyces verticillus. In this occurs as copper chelates with the ligands, and that will be provided by pyrimidine, amide or amine function groups. This is for natural bleomycin. Here the copper, no, that is removed from the pharmaceutical form, but this metal chelate, no, that is the key to anti-tumor activity. Inside the cell, this bleomycin forms chelate with Fe2 and molecular oxygen, and we are getting one complex that gives rise to hydroxy radical and superoxide radical. These radicals degrade the DNA by reacting with it. We will see the uses. This is used in the treatment of carcinoma of cervix, head and neck, larynx, penis, skin, testis, Hodgkin's and non-Hodgkin's lymphoma. So here today we have seen two antibiotics, one is anthracyclines and another one is bleomycin. The left three antibiotics we will see in next class.